Thank you so much, Kat, for the introduction. So I'll be telling you all today about this phenomenon that we're calling animal methods bias. I'm going to start out with going over briefly some of the limitations of animal-based methods. Then I hope to finally answer this question that you may be wondering, what exactly is animal methods bias? Then we'll dive a little bit deeper and explore what are some of the consequences of animal methods bias. And finally, I'll tell you about some of our ongoing mitigation efforts. I'd like to first ground this discussion today in the devastating effects of animal experimentation on the animals themselves. There are many scientific reasons to avoid using animals for biomedical research and testing, which I'll go over in just a moment. But there are ethical reasons as well, and sometimes these reasons can get overlooked or taken for granted. So as we explore animal methods bias today, I challenge all of you to think not only about the scientific implications of a system that prefers using animals or is biased toward animal-based methods, but also the ethical implications of such a system and the impacts that this has on these precious animals. So to get into some of the scientific reasons for avoiding animal-based methods, we often hear about the crisis of translation. On the journey from bench to bedside, 95% of drugs trying to overcome this translational gap fail in clinical trials. And this lack of translation can be attributed to many different factors. But one of the major culprits is the use of animals in preclinical research and the limitations of animal methods. The limitations of animal experiments often focus on internal validity, the scientific robustness of a study's design, conduct, analysis, and reporting. Issues with animal research often include small sample sizes, low rates of random allocation, and incomplete reporting. And some of these problems can be overcome, and many argue that once they are overcome, the crisis of translation will be solved. But as Pandora Pound and Meryl Ritsky-Heutige argue in this really great paper from 2018, better rates of translation will never improve without better experimental external validity, the representativeness of a model, and its ability to mimic the complex human biology and health. They argue that the external validity of animal models is ultimately what makes them bound to fail. When animal models fail to reliably predict clinical safety and efficacy, it's not just a problem with the design and the reporting of animal experiments, but the choice to use animals as a model for humans in the first place. Non-animal-based methods have advanced a great deal over the last decade, and this calls into question the status quo of animals as the go-to for studying complex biological processes and the mechanisms of health and disease. These methods have improved on the limitations of animal-based methods, but their ability to replicate the molecular, cellular, and physiological mechanisms of human diseases more reliably, and to translate preclinical findings into safe and effective treatments for patients in a more predictive manner. 3D culture systems like tissue chips and organoids can do this in a really elegant way. They can be used in a variety of applications like developmental biology, infectious diseases, rare genetic disorders, drug testing, and even to enable precision medicine. And they can do this by modeling a wide variety of human tissues, organs, and body systems. This is a graphic from a 2020 review on organoids that shows some of the many tissues that organoids have been used to models. Non-animal-based methods like tissue chips and organoids are powerful tools that can replace the use of animals in many applications. But there are many barriers to their uptake, including the publishing peer review process. As you all know, publishing plays a crucial role in the advancement of science. It's this pivotal step in the journey from bench to bedside. It helps implement medical interventions, and it also plays a role in the progress of researchers' careers. But this publishing process can be influenced by biases that are introduced both on the side of authors and on the side of journal editors and reviewers. According to the Catalog of Bias, which is a database created by the Center for Evidence-Based Medicine at Oxford University, publication bias is defined as when the likelihood of a study being published is affected by the findings of the study. But a couple of years ago, my team and I started to wonder, what if the likelihood of a study being published is affected by the methods of the study, namely animal versus non-animal methods? Recently, early evidence of this, a newly defined type of publication bias, has emerged. In 2021, Donald Ingber published a paper, "Is it time for reviewer 3 to request human organ chip experiments instead of animal validation studies?" In it, he questions why animal data is still considered the gold standard in human health research. And he presents evidence that organ chips may better suit this purpose. But notice how he frames this as a problem with peer reviewers. And this phenomenon defines this emerging type of publication bias, in which editorial policy or peer reviewers, or even editors sometimes, request that animal data is provided to validate studies that were produced using human biology-based approaches, like organoids or organ chips. And sometimes they do this as a condition for publication. Thus, we've defined animal methods bias in publishing as a preference for animal-based methods where they may not be necessary, or where non-animal-based methods may already be suitable, which affects the likelihood of a manuscript being accepted for publication or introduces a significant delay to manuscript acceptance. Animal methods bias may affect other aspects of the research enterprise as well, like the review of grant applications. But so far, we've mostly been concerned with animal methods bias in publishing. And this is a big problem. It may lead to scientists performing animal experiments in haste, not because they initially designed their study that way, but just because they want to get their paper accepted. And these additional requested animal experiments may lack rigor or justification, and they stand in the way of the uptake of more ethical and physiologically relevant human-based methods, like tissue chips and organoids. So, in collaboration with the Humane Society International, we set out to understand when and why this occurs, with the hopes of using this evidence to inform solutions and to urge publishers to take action. In spring of 2021, we set out to gather evidence by conducting a survey to assess the experiences and perceptions of authors and reviewers related to animal and non-animal-based experiments during peer review. The survey contained 33 questions and was housed on the SurveyMonkey platform, and we disseminated it using a URL link via research and technology networks on LinkedIn, through Physicians Committee doctor and researcher email lists, through allied group newsletters, and our own personal networks. At the top of the survey, we defined these terms to avoid any confusion. An animal-based experiment we defined as an experiment performed in a living non-human animal or in a non-human animal-derived organ, tissue, or other biological product. So for example, an animal in vivo model or an animal cell-derived in vitro model. And we define non-animal-based experiment as an experiment performed in a living human or in a human-derived organ, tissue, or other biological product, or in a non-animal specimen or in silico. So for example, a human cell-derived in vitro model, even if it uses animal-based materials like buffers or antibodies. The demographics of our survey respondents were that there were 90 respondents total, and after exclusion criteria were applied, there were 68. They represented a broad range of biomedical and related fields, primarily coming from the academic sector, slightly skewed toward female respondents, and representing a broad range of countries, but the USA was most well represented. They primarily held research doctorates and some held medical doctorates, and they were fairly evenly distributed across levels of seniority based on years since graduation of their latest degree. We asked survey respondents, how often do you perform animal-based experiments for the sole purpose of anticipating reviewer requests for them? As in, you did not think the experiments were necessary outside the context of review. 41 respondents indicated that they never use animals in their research for the sole purpose of anticipating reviewer requests for them. But 21 respondents said that they do this rarely, sometimes, or often. We then asked, during manuscript submission peer review, how many times have you been asked for animal experimental data to be added to a study that otherwise had no animal-based experiments? 31 respondents indicated that they've never been asked, but 30 respondents indicated that this has happened to them at least once. When asked whether they thought these requests for additional animal experiments were justified, of those 30 people, only 3 indicated that they felt the request was justified, while 14 indicated that it was sometimes justified, and 11 indicated that it was not justified. When asked to elaborate, here's what the respondents said. Reviewers ask for animals out of habit. The study was about heterogeneity of cancer cells from human tissue samples. It was irrelevant to do an experiment on mice. Referees ask for animal experiments because it's customary, not because it's necessary. Many are unaware about the potential of in vitro and in silico methods. The journals that wanted animal experiments were journals with higher impact factors. People who do animal experiments are perceived to be better scientists because they are allowed or encouraged to submit to those journals with higher impact factors. There's no incentive to not do animal experiments. In my experience, reviewers on grant panels are even more likely to ask for animal experiments than manuscript reviewers. The survey identified potential sources of animal methods bias as reviewers' preference for animal methods or their lack of awareness of appropriate non-animal methods for a given hypothesis. And it identified potential consequences of animal methods bias as delayed time to publication, rejection or withdrawal of the manuscript, publishing in lower impact journals, as we heard from that survey respondent just now, and performing additional animal experiments which would have not otherwise been performed. The sample size of this survey was too small to be representative of the population, so we're unable to determine the prevalence of these requests for animal experiments by reviewers. But this survey did provide preliminary evidence of animal methods bias in publishing, and it has served as the foundation of our work to further explore this issue and to develop strategies to mitigate it. If you're interested in reading more, there's a preprint up on bioRxiv, and the paper is currently under review at Altex. So after an August 2021 presentation about this issue and the survey at the World Congress on Alternatives and Animal Use in the Life Sciences, I assembled a steering committee to organize a workshop to address animal methods bias in scientific publishing. In April 2022, we gathered diverse stakeholders virtually on April 20th and 21st for 3 hours each day to carry out the following charge. To explore a range of stakeholder perspectives, including from academic and industry researchers, journal editors, government representatives, and advocates. To describe the current state of animal and non-animal-based experimental systems, to describe animal methods bias in publishing and related biases in publishing and peer review, and to identify potential causes, consequences, and potential mitigation strategies for animal methods bias in publishing. Our workshop speakers are listed here, and they illustrated the evidence for this problem and the current state of non-animal methods and their suitability for answering many research questions. They provided information on trends for the publication of animal versus non-animal methods. They described the responsibilities of journal editors, different models for scientific publishing, including open access and different financial incentives, as well as authors' experience with publishing, lessons from transdisciplinary research and systematic reviews, and they discussed the multifactorial nature of non-animal methods uptake. Conversation among participants took place at the conclusion of each day's talks and was facilitated by prompts from workshop organizers using the Mural platform. And this is just a screenshot from one of the breakout rooms using the Mural platform to brainstorm. Some barriers to addressing animal methods bias that were identified by workshop participants were the high-pressure nature of the research environment, impact factors again, financial stakes, animals as the quote-unquote gold standard, institutional inertia, and a lack of knowledge or a desire to learn about non-animal methods. Workshop participants came up with many different recommendations for mitigating animal methods bias, but I'm just going to go over a few of these that I think are most relevant for this webinar. So first, in the scientific community, we recommend to build awareness about animal methods bias through things like conference talks or webinars like this one. Also, to increase authors' confidence in their ability to challenge reviewers' requests for animal experiments. And I'll be mentioning this one again in just a few moments as we talk about our ongoing work. We recommend for journals and publishers to encourage the pre-registration of studies and to encourage thorough reporting of methods and results, including negative results. We recommend funders, governments, and policymakers to prioritize funding for non-animal methods, including for training and infrastructure, and to advance the validation, standardization, and reporting of non-animal methods. You can read more about the workshop in detail in our recently published report in all texts. After the workshop, several attendees and members of the steering committee came together to form an animal methods bias task force that's committed to continuing to explore and address this issue. Within the task force, the evidence building working group is gathering additional data on animal methods bias in scientific publishing through a follow-up survey to the one that I described at the beginning of this talk. We've been working with a US-based data-focused animal advocacy organization called Faunalytics to redesign the survey questions and determine a study participant recruitment plan in a rigorous manner. And our hope is to get enough respondents that we can make an estimate of the prevalence of authors' experiences with animal methods bias, and then to use this evidence to bolster our recommendations for journals, funding agencies, and other important stakeholders. Also in the evidence working group, we're analyzing publication data by comparing final published articles and their corresponding preprints to look for animal experiments that show up in the published article but were absent from the preprint and thus likely originated during the peer review process but weren't included in the original design of the study and therefore may have been requested by reviewers, editors, or other influences. Elsewhere in the task force, the mitigation working group is planning to use the evidence gathered from the surveys to work with journal editors and publishers to improve policies. And we're developing educational materials and support for authors who wish to avoid animal methods bias and then to address it if it does happen to them. These guidance includes these elements here. So we're working on preventing animal methods bias by developing guidelines on pre-registering non-animal studies, providing a directory of human-based models and data sets that researchers can use, and guidelines on preparing non-animal study manuscripts. During the submission process, we're providing a directory of non-animal method-friendly journals as well as guidelines for finding reviewers to suggest with submission. During the review, if authors happen to get back a biased review, we're providing guidelines and templates for responding to biased reviewer comments. And we've also created a bibliography of references on non-animal methods, translation, animal welfare policy and legislation in different regions of the world, bias and peer review, and several other topics. And lastly, we're intending to create a platform for community support for researchers to form collaborations, to share experiences they've had, even maybe to share communications they've had with reviewers and other things. So lastly, I just would like to acknowledge the many people who have contributed to this work. It's been a really exciting collaboration between lots of different organizations and researchers and advocates, some of whom are listed here. So thank you very much. And I guess we can get to the Q&A portion.
Thank you very much, Catherine. I also put in the chat that people can share their own experiences. Maybe we have time for that. If there are people in the audience who had similar problems, let's put it like that they were asked to include animal studies. The first question is about the report of the survey. Is it publicly available? Because this person is a member of the OnTalks consortium, and they are aiming for full implementation of NAMs and probabilistic risk assessment, and it would be very valuable to have an insight in this report.
Yeah, so the original survey manuscript is up on bioRxiv. You can just Google a survey to assess animal methods bias in scientific publishing. And the manuscript is under review at Alltext. So hopefully in the next few weeks or months, the final publication will be up there. But yes, the preprint is available for you to explore now.
And you put everything in you made all the questions and everything available. That's great. Okay.
Yeah. So the survey questions are in the supplement, I believe. And the second survey that I mentioned, the follow-up survey, we are still in the study design portion. We're almost ready to launch, but we're hoping to launch that second survey sometime in the next couple months as well. And that one, we're hoping to have a lot higher of a sample size. And we've also, by partnering with this organization, Faunalytics, we're hoping that the design of the research questions will be much more rigorous, and that the results of that follow-up survey will really be a lot more convincing for different stakeholders like journals, and that we can use that to, try to implement some sort of journal policies or changes, with funding agencies.
Now we have a very long, a short question and a long answer, and I will read it to you. The question is, why do they still use animal testing if the non-animal methods are so much better, is the question. And the answer the person gives is that the pharmaceutical industry wants to get as many new drugs to clinical trials as possible, so as to increase the number of drugs that they can put on the market. Using nouns such as human organs on a chip would severely reduce the number of drugs entering clinical trials because of early detection of human adverse drug reactions. This is not good for business, the person says. They're aware the pharmaceutical industry thinks it has found a blockbuster drug. That means a new drug that promises to make lots of profits because it can be prescribed to a large proportion of the population. Then it must certainly it most certainly does not want to test via human-based research technologies in case the drug is shown to cause toxicity in humans. Bringing a new drug to the market costs billions of pounds and can take over a decade to develop. These high monetary and time investments are both strong contributors to today's skyrocketing healthcare costs and sign. That was the comment. I don't know what your thoughts are on this, because it doesn't seem to make sense to me if they produce something they can't get marketed then.
Yeah, so I think what this person was trying to say is that if we use methods during preclinical testing that are more predictive of human outcomes, that more drugs are going to fail in the preclinical stage. And so companies don't have this opportunity to put that drug on the market. And I don't know, I think that's a pretty cynical view, but I do think that, part of the barriers to non-animal method uptake are financial. And there are companies who have a financial interest in the continued use of animal methods in preclinical research. There are companies that sell animals to researchers across the globe. Obviously drug development companies are worried about their bottom line, but you would think that they would think that drug development companies, because the bottom line, their profit, is so important to them, you would think, or I would think, that having preclinical methods that are more predictive of outcomes in clinical trials would be beneficial to them, so that then those are the drugs that pass the preclinical testing also pass the clinical trial testing, and then they can go on to have a more successful launch of that drug. But I think finances are only one part of the picture. There's also, and I mentioned one of the barriers being institutional inertia. And this is sort of just the fact that the use of animal methods, it's been the gold standard for decades. And for many of these PIs who were trained using animal methods, that's what they know. And so that's what they teach their students, and that's what gets proliferated. And so that's why we think a lot of the solutions really lie within educational institutions and also funding agencies. We need to be putting public funds toward research to develop and disseminate non-animal methods, and to really to train the next generation of scientists, and to put infrastructure in place that makes using non-animal methods and accessing them easier. These companies that sell animals for research, we need just as many or more companies that are providing non-animal methods to researchers and drug companies.
Also, when I read this question or this comment, I thought that doesn't make sense to me, because it's, I mean, obviously the preclinical part is mostly done, or often done, in academia, and then if it looks successful, then they pick it up, because it is all about not wasting money on the wrong things. So I think that the drug companies don't have the big interest in testing all those drugs, and then they fail because of toxicity or lack of efficacy. So the next question. And maybe one other thing. I don't know what your thoughts are on that, but I thought it's very interesting to see that many animal studies run in parallel to human clinical trials, which doesn't make any sense, because you think that humans should be protected by having using other animals first. But that's actually not the case. And that kind of also implies that actually maybe those preclinical studies don't even inform the clinical trials, which is, it's a whole different problem. Obviously, you touched on that, but there's also a problem of quality of those studies and so on and so forth. I don't know if you want to comment on this whole weird parallel world that's going on.
Yeah, that's a good point. We have seen, like we saw it with COVID, with some of the, testing for COVID treatments or prophylactics, that they would begin clinical trials and concurrently run some of the preclinical animal tests. But it sort of shows, I think, like you said, that if these methods aren't really being used to protect clinical trial participants anyway, or if we're acknowledging that they aren't predicting clinical trial outcomes anyway, then what are we really using them for? And are they necessary? Or are there other methods like organoids or tissue chips that we can use to better predict safety in humans and effectiveness?
Yeah, maybe since you are US based and I'm more Europe based these days, it was very exciting for many people, and including many from the general public, to hear the plans of the FDA about allowing the NAMs, the non-animal methods, for testing. Can you elaborate a little bit on this to give our listeners a little bit of an idea what's happening? And also, obviously, it sounds better than it is, but it is important. Maybe you can say a little bit about that as well.
So you're talking about the FDA Modernization Act. Well, I wish I could answer this question, but truth be told, it's my colleagues who have really been working hard on this. So I'm not as familiar. I'm mostly focused on what happens at the NIH. There was a bill that was signed into law to modernize the FDA. The aim was to basically remove the requirement that new drugs have to be tested in animals. But yeah, I don't really want to go further because I think some of my colleagues would be better at. No.
That was great news here everywhere. Like all the animal advocates were very excited about this. But I read many commentaries on that, and obviously it doesn't automatically mean that everything will change from today to tomorrow. Like it's still a process, but at least it opens up for for the NAMs. So another question from a listener is: do you think that a measure to avoid or reduce animal methods bias in publication or in publishing could be the journal editors to set a minimum of knowledge or criteria for reviewers to fulfill?
So I think that I'm not sure if this answers what this person was asking, but one of the things that we're hoping to do, we hope to engage with journals and editors and reviewers to educate them, to raise awareness about this issue, but maybe even to provide some sort of training. Like we've seen, one of the ways of mitigating bias that you'll see is bias trainings. And so sometimes these are like a training module. You watch some scenarios where an interaction is biased or some sort of biased comments are provided. And then the training will go into detail about sort of what's going on here. Why is this interaction biased? And how can we avoid this bias? So that's sort of one of our ideas for, educating reviewers and editors so that they understand, if if the point that you're trying to make in your review is that more experiments need to be done to bolster the evidence that you're presenting in your findings, that instinct to recommend an animal study or an animal experiment is not the gold standard anymore, that there may be other ways of testing this hypothesis or validating. There are not only methods like in vitro methods like organoids and organ chips, but in human health research, the gold standard is human health outcomes and human health, biological mechanisms and biomarkers. And so maybe there's some sort of clinical data that you can provide to really validate your findings or somehow, bolster the evidence that the claims that you're trying to make. So I'm not sure if I answered that question, but we are trying to pursue these kind of like education, awareness building, or even some type of training module or guidelines for reviewers to hopefully mitigate this bias.
And can you once repeat the definition of animal methods bias? Because there was a comment and question in the chat where it becomes clear, I think, that I can read it to you. So the question is that, of course, nobody wants to be criticized by reviewers, and obviously that's not, your first reaction is like, And then the person asks, how are you going to differentiate in a future study between requests of animal studies due to bias versus due to scientifically reasonable needs? And I think you already had this in your definition that, but.
Yeah, so we define animal methods bias in publishing as a preference for animal-based methods where they may not be necessary or where non-animal-based methods may be suitable, which affects the likelihood of a manuscript being accepted for publication. So the distinction is that this is a request for an animal experiment where it's not necessary, where there are other methods that may exist. And I think that this person is making a good point, which is, and we've considered this a lot, which is like, should reviewers be recommending these kinds of major shifts in the design of your study? And actually, this also makes me think of one of our recommendations that I mentioned is to pre-register your studies. And our idea behind recommending pre-registration is that you design your study, you lay out your plan, you lay out, your plan for your experiments and how you're going to analyze the data. And then you put that into some sort of study registry, or even, publish it as a registered report. Many journals are now offering registered report article types, which would then undergo a peer review. And so when you're publishing your final manuscript, then there's evidence that pre-registration can increase acceptance rates. And our hope is that what it can also do is prevent reviewers from recommending these kinds of major changes to your original study design, like you need to do a whole nother type of experiment or provide more evidence. Some of the recommendations that I didn't go over that came out of the workshop were greater transparency in peer review and publication. So there are some, there's the open science movement now to make your methods fully transparent, to report them to the extent that your study and your findings can be reproduced by another research group. But also in the context of peer review, there are levels of transparency in peer review. So sometimes you can, have a peer reviewer's name be known. You can actually have the peer review comments or reports be published alongside the publication. And you can even, there are some models now that allow reviews to be provided after the publication gets accepted. So members of the community can further provide their review of your paper, and you can continue to revise your manuscript accordingly. So yeah, that's a really definitely a big component of this, is sort of what are different ways that we can conceive of the peer review model to improve it that can help this bias.
And a comment, when I was a member of an animal ethics committee, I tried to argue against the use of certain behavioral tests, since they are very stressful for the animals and do not really answer the research question. For example, the use of the forced swim test. Researchers sometimes argued they needed to use a certain behavioral test because the journals require it, despite not being able to come up with convincing argument for how to test, and how the test would answer the research question. So unfortunately, it's not only a problem about non-animal methods, but also which animal tests the journals require.
Yeah, this is a really good point. I wasn't aware of journals that require tests like the forced swim test, but I am aware of journals that in the scope on their about page, they explicitly require that studies, that manuscripts for submission have some sort of animal in vivo component. So we definitely know that there are journal policies that also, proliferate this bias. And that's one of the things that we are hoping to do in the mitigation efforts, to maybe get some of these journals to change their policies that require certain animal experiments or, in vivo evidence or specific animal experiments like a forced swim test. But I do this is a really great point that, the bias isn't manifesting just as like one person requesting that you add an animal experiment like during the peer review. This is a much bigger issue. Sometimes it's baked into journal policy. And it also, I mentioned earlier, like the funders and policymakers play a role in this as well. So addressing this issue, right now we're sort of focusing on publishing and the peer review process, but this is such a bigger systemic issue that solutions are really going to need to be happening in many different areas.
Yeah, I would already be happy if we just would agree on some animal models that are obviously not translatable. I mean, there's a number of them that we could just stop using, but they are still funded, they are still published. This is kind of shocking, because that's, you wonder how we will ever make any change, or there will be any change. So there's a word of encouragement, which is great. They published a pair of articles in the JACC Basic Translational Science regarding the ineffectiveness of animal studies in human pharmacology research. It became one of the highest downloaded articles from the journal, to the editor's surprise, and led to a number of webinars in animal research issues, Elsevier publications, as well as an offer to publish a textbook on this issue. So that sounds wonderful. Regarding what the FDA just changed, the person is asking if you think that this can impact animal experimentation in general, as you talked about the inertia of the sector. It's so big, and they ask, can this help reviewers and journals to change their minds about the so-called gold standard?
I do think that one of the big barriers has been in the US the fact that to a large extent, animal tests have been required for a new drug entering the market, or even cosmetics. And so removing this barrier, I think, definitely has effects throughout the research enterprise. And it is one of the areas that we can point to when we're engaging with journals and saying, maybe you need to change your policies or just make your reviewers aware that animals aren't the gold standard anymore. We can point to this type of thing, like the FDA is no longer requiring them. And, definitely, I think, like I said, it's a systemic issue, and a win in one area can have downstream effects in other areas.
I've got 2 questions. Is there any drug testing or any other kind of testing that you would recommend that it should be done through animal testing? And the second one, according to you, is there any kind of ethical animal testing? If there is, could you elaborate on why it would count as ethical?
I can't speak to any specific drug testing that I would explicitly recommend use in animal. Is there any kind of ethical animal testing? I mean, this is it's a really hard question. I think that, what I would like to bring the focus on is, maybe there's not a replacement right now for every single animal experiment or animal model, but we're certainly never going to get to the point where we can replace animals in research if we don't invest in non-animal methods, if we don't invest in training the next generation of scientists, if we don't invest in infrastructure, like I mentioned earlier, if we have, models available for use for researchers to use, if we fund more research, to develop these models and to use them to study a variety of applications, then we're never going to get to the point. And so this question of what is an ethical animal test? I think it sort of is, there's no good answer to that, ultimately it involves treating a sentient creature really horribly. And if we can avoid that, then we should. I think we have an obligation to do that. We have an obligation to pursue any kind of method that can potentially prevent that.
In OnTalks, they are reaching out to all relevant stakeholders, regulatory authorities, industry, and NGOs in relation to their ambition in moving towards NAMs and probabilistic risk assessment, and thereby to have a more safe world. And then there is a question coming. And in OnTalks, we experienced that the industry is not the strongest opponent against NAMs. However, they don't want to invest in NAMs if the tests are not accepted by the regulatory authorities. Regulatory authorities are biased of traditional testing, or they are biased towards that. And there's a need of dissemination to this target audience, even with reference to the fact that OnTalks is one of the EU-funded projects aiming for NAMs and GRA. There is reluctance in some agencies, for example, ECHA, where EFSA is slightly more positive. The question is, how can Physicians Committee put more pressure on relevant authorities to stop comparing animal testing with NAMs, and how to secure validation to secure proper confidence in NAMs?
That's a great question. That is certainly one of the things that I and my colleagues work on, which is to put pressure on regulatory bodies, on funding agencies, on policymakers, lawmakers, these really high-powered stakeholders. It's a good point to stop comparing animal tests with NAMs. And I think it's also a really good point too, that we do need to secure validation and proper confidence. And I mentioned in my talk that one of our recommendations is to really focus on validating non-animal methods, standardizing, benchmarking, and then obviously reporting. There are some people now who are developing reporting guidelines for in vitro methods. So just as, the internal validity issues with animal methods have been a big problem, it's also it's a problem with any kind of experiment, but we also see it with in vitro methods. And so we really need to get these things in line if we're going to build trust in non-animal methods and really make that shift. We need to make sure that we're confident and I think that really, pressuring these, regulatory bodies and funding agencies is a big part of it. But it takes time and money. And so I think that's sort of the big question. But, things like, this, where we have, scientists like yourselves, I think it's just as important for scientists to be also expressing your views about why these things are important. The scientific community is a major stakeholder as well. And so we need more scientists to advocate for change.
So let's see, we have many more comments. For example, one person says some reviewers don't have a clue about non-animal methods. They reject papers as they do not understand them, especially the mathematics, if mathematics is involved. So that's another experience to share here. Another question, the person says, I'm meeting with senior staff at Emulate tomorrow and will be addressing the exact issue you were just discussing, that of NAMs being developed as adjuncts rather than replacements of animal models, as these technologies are implemented and regulators get up to speed on validation and acceptance of these methods. I have been on several webinars with industry that project everywhere from a few years for a few decades. Happy to follow up as I learn more.
We have seen this actually in the States. The NIH just in December launched a working group as a part of the advisory committee to the director to assess areas for investment in non-animal methods, which is really exciting. And we're counting this as a win. The NIH is considering where to invest in non-animal methods, which is something they've been really putting off and fighting for a long time. So it's really promising. But it is interesting because the way that they frame this is that they're insistent that non-animal methods are complementary to animal models. And it is frustrating. And I think that, part of what we're doing is really trying to convince the NIH and, other stakeholders that it doesn't have to be complementary, that it can be a replacement if we can get the methods, if we can build trust and get them accepted, then it can be a replacement. But still, I think that I would count it as a win, regardless of how it's framed. If we're getting investment in more non-animal methods, if we're getting more non-animal methods accepted by regulatory agencies, these are really the first steps toward replacement. So I still count this stuff as a win. But, I think part of our job is to challenge this notion that it's complementary, that we'll never fully replace animals. I think it's sort of just the way I see it is that sort of perspective just lacks imagination. And it doesn't consider, what I talked about at the very beginning of my talk. What underpins all of this is, the animal ethics, and these animals deserve for us to have more of an imagination about what is possible in terms of replacement. So
there's another comment, that's something we definitely have to deal with. Basic and applied research always, or I would say mostly, insist on an animal for the full biology, especially in universities in the UK. The person says holding back all the students as their qualifications will be outdated. I guess that's a similar issue you have in the US and everywhere, right?
Yeah, I mean, so first of all, I do think that, these new methods are getting better and better every day in their capabilities. And so, like you said, organ chips, we do see now organ chips that are configured with multiple different cell types and can recapitulate different organs and body systems interacting with each other. And so, you can replicate to a certain extent full body, full organism, or at least partly the physiological interaction between different organs and tissue types. But I think that the key here is that this is about training. And this is why I'm always driving home the point that, we need to be thinking about how to train the next generation of scientists and, thinking about the ways that the methods we've always been using are outdated. And I think we have to be frank about, like I said, part of the institutional inertia is just that a lot of these PIs, that's what they trained in. A lot of these professors, these are the methods that they trained in. And so, of course, that's what they're training to the students who are working under them. But I think this is where funders come in, because if funders are explicitly providing funds for training in newer modern technologies and methods, then the PIs will be applying for those grants and they can, learn themselves some of these new methods. So, I appreciate that characterization. And I do think that, this question of training is really important.
Another person is asking, why is the fact that NAMs are generally less expensive than animal experiments, apart from also being more accurate and providing faster results, not more of an incentive to use NAMs instead of animal experimentation?
Yeah, I don't know. I mean, I agree with, I think what this person is trying to say is that the question of cost is really important, and we should be clear about that, that animal experiments are really expensive. They take a lot of time. We have to feed the animals. We have to house them. We need to have veterinarians. And so you save time and money when you use some of these non-animal methods. So absolutely, I agree. This is one of the major selling points.
However, I mean, I don't, it would be interesting to look into more into detail in this, because obviously there are some of the new methods that are very expensive. And I mean, obviously, because they're not maybe also produced in such a large scale yet. So I think costs would come down more if we switch more to those. But it would be nice to see some calculations on how much money is spent. Because I just know from the US, it's very hard to filter from the proposals how much is in vitro, in silico, and versus animal work. So I think, you know, for when you look at NIH, what they fund, for example, and it's the same in Europe. You cannot really, because obviously many of the animal studies also have some in vitro work involved and other things. But yeah, I think the cost issue would be interesting to look into more closely. I think it's a little bit more complicated. And obviously, the other thing that is kind of frustrating is the distribution of funding at the moment. I can just say from what I see, it's just there's not enough funding for the NAMs. It's easier to get funding for animal-based research. And that obviously has a whole cascade of events that follow, because then obviously journals are more used to animal studies. Everybody's more used to that, and that's just the more accepted thing. But it's actually, I don't know, there were ideas about how to distribute or like change the distribution of the money. But obviously there's a lot at stake, and obviously there's a lot of opposition from the people who currently receive this money. But I think that academia could do a better job in being as open-minded as you would think they are, you know, because it's not, I don't know about your experience with academia, but I'm always astonished how locked in about this institutional lock-in that you find in academia, maybe even more than in industry, because I think industry is more money-driven, and then they are more minded towards what works, you know. But,
right.
Yeah. So.
Yeah. Yeah, and I think that I think that this principle of like cost savings, it may not be true for every single application right now. But, you know, every new technology requires some sort of investment. And I think it takes time to see a return on that investment. And so, of course, transitioning away from animals is going to cost a lot of money. But I do firmly believe that down the line, we will see cost savings. Just in terms of, you know, these methods being more predictive, then we're performing less experiments that are wasting money because they don't predict outcomes in humans.
Another comment is never underestimate the power of the dollar, not just in private industry, but in funding availability. People in academia will follow the money as well. Their careers depend on bringing in research dollars to universities. So as we are taking a multi-pronged approach to completely transforming the approach to life science research, it is critical to find the money to support the change. Investors in biotech, investors from biotech in training, and investors in scientific research ethics will not drive the transition quickly enough.
Yeah, I mean, there are some resources like, you know, I mentioned that NIH is thinking about how to invest more in non-animal methods. But right now, it's definitely greatly overshadowed by the investment in animal methods. But then there are also, you know, other private funders and you know, private foundations that fund this kind of research. I don't know if there's like one place that folks can go to look to, but yeah, I agree with this. You know, we need more funding. And I think that the NIH, as the largest single funder of biomedical research in the world, I think has to be one of the obvious solutions to finding more money. With a yearly budget of $50 billion, you know, if we can put a fraction of that toward the development and use of non-animal methods, I think we'll be in a much better place. And so that's sort of consistently what I'm trying to do, is to convince them to do that. And they're sort of slowly starting to change, but yeah, funding is a major hurdle.
Yeah, indeed. So there were some ideas about having some kind of an overhead. If you do animal experiments, you have to pay certain amount of money into another pot where you then can fund animal-free methods. But I think that was an idea of somebody I spoke to who would have had the power to do that, but then was unwilling because of the backlash. So I think it's very difficult to start that. But I mean, we have to start somewhere. I don't know. There's another comment in the Q&A about obviously that about time saving with NAMs, that it's not always that easy because it depends what you're doing. And here for original organoids, it's of reasonable quality. It takes at least 2 years with daily medium change, the person says. And then for brain organoids, he says it's easier. Obviously, nobody claimed that this is easy. I don't think, and also that it doesn't. I think it's a huge challenge, and we are still in infancy with many of those new methods. But I think they look promising, most of them, and so let's hope that we can push them further. And obviously, that's what all those areas you mentioned. There are many areas where we have to make a change to further this whole transition.
So I don't mean to minimize, these challenges and just make a blanket statement about, the cost savings and time savings. I think that, one of the things that we're promoting is changes in infrastructure to make these non-animal methods more accessible. Creating a particular type of organoid from scratch obviously takes a very long time and is very challenging. But, and if you can just order a mouse from a catalog and have it there within a week, obviously that is going to be a lot easier and a lot faster. But imagine if you could order that organoid from a catalog. That's sort of like what we envision for the future. And I think that's only possible if we invest.
Yeah, totally. I totally agree. Because I mean, the other thing that's super difficult, and that's a whole different talk. Actually, we had this talk last year. We need tissue banks. We need all this human tissue from diseased patients and of healthy patients and so on for those new methods. And there's a whole ethical other problem, like, the ethics agreements you need and all those things. And in most university hospitals, there's not a good system in place to ensure all that, including quality and timeliness and all those things. So there are many problems, but I think you made this very clear in the beginning. I mean, we are only getting so far with using animal models, and not far enough for most diseases we need cures for. So I think hopefully we can agree on that, that it's worthwhile just for science to look for human relevant methods.